Welcome to today's Chemi colloquium. I invited my good friend Timm Krüger a while ago before it became apparent that there was no possibility for him to actually visit the department, unfortunately. So he's kindly agreed to give an online talk for us, but I plan to still invite him to visit us in the future. Timm is a reader that is equivalent to associate professor in chemical engineering in the School of Engineering at the University of Edinburgh. He specializes in lattice-Boltzmann modeling and simulation of complex fluids. In 2018, he was the recipient of an ERC starting grant for lattice-Boltzmann applications to microfluidics. And he's applying this method to emulsions, suspensions of deformable particles, including cells. And today he will tell us about blood flow modeling in the human body from the cellular towards the organ. Timm, the floor is yours. Please share your screen.
Thank you very much for inviting me. And Valeria, it's great to see you again after some time. It's a pity that I cannot be here in person. And by here, I mean Delft. But we will definitely do that very soon as we can. Right. Thank you very much. I think, let me see if I can move this somewhere else. Okay. I think I have about half an hour. Is that correct? Perfect. I know that everyone is suffering from Zoom fatigue and Microsoft Teams and Skype fatigue and whatever. So let me make it as brief as possible. Thanks again for the invitation. I will talk about blood flow modeling in the human body. In particular, I want to emphasize how we can go from the cellular scale, which means the scale of a single, let's say, red blood cell, up to the organ scale. And I will not talk much about the computational methods, but try to give you a general overview of what is happening and what our ideas are. What do we have here? Okay, so here you see a nice picture of Edinburgh. It's probably one of these days you have only once a year in Scotland. And if you see the picture behind me, some people ask me whether it's a battlefield. It's actually not. It's people playing golf. And as you know, golf has been invented in Scotland. It's actually a very nice country to live in. And if you haven't been here, then I strongly suggest you pay a visit once it's possible again. A very, very quick overview of what my group is focusing on. There is microfluidics and blood flow, but there's of course overlap between microfluidics and blood flow, and also complex flows. Complex flows is something I would say I define it as fluid flow where you have additional physics. So it could be in complex geometries, it could be with surface tension, some kind of maybe magnetic fields, anything that is making the problem a bit more complex. So everything that is not blood flow or microfluidics, I just put into the complex flows column. But what I want to focus on today a bit more is actually blood flow, and one important aspect here is how is it related to health and disease, and what can we learn from that? The outline of my presentation is that I will give you a one-slide summary after the introduction of the numerical model. I think it's only one slide, and then two applications that we have recently published or at least submitted. One of them red blood cells in bifurcation, so the other one red blood cells in networks. And after that, of course, an outlook and a summary. So let's start. Why is it all important? What you see here is an example of blood flow in health and in disease. At the bottom left, you can see a snapshot of my own retina, probably taken about a year ago when I went for an eye check. And I saw this image on the display, and I said, oh, I need that image. Can you please send it to me? I want to use it in my research presentations. So that is actually a healthy retina, I was told, which is fantastic. But on the right-hand side, the image you can see here, the illustration shows a solid, it shows a solid tumor growing, and when this tumor is growing, obviously it needs some space, and as a consequence, blood vessels can be affected by that. So blood vessels can be compressed by the growth of tumors, and on top of that, tumors are creating some kind of unstructured microfluidic environment where blood vessels form, new blood vessels form, and they create some kind of network that is not well structured. And as a consequence, you have, for example, shunts between arteries and veins, and you have a, well, unhealthy network of blood vessels. And why is this all important? You can imagine that a healthy blood vessel network has been optimized for various purposes, in particular oxygen delivery to the tissue. And in a tumor, all these nice regulation processes that lead to a nice layout of the network, these processes are broken or they don't work that well, and you get quite messy geometry of the blood vessels. And as a consequence, you typically have regions in the blood vessel network within solid tumors in particular that are less well oxygenated. And you may now think, okay, if you have a tumor that is less oxygenated, that should actually be good, because tumors, they will suffer when they have less oxygen, so they should be less dangerous. But unfortunately, that's not the case. Tumors are usually getting more aggressive when they are under stress, and in this case, chemical stress, so they don't have enough oxygen. And also it has been shown that the treatment of tumors is detrimentally affected when the oxygenation levels are lower. So for example, a tumor that is well oxygenated is much more susceptible to radiation treatment than a tumor that is not well oxygenated. And therefore the strategy is actually not to deplete a tumor of oxygen, but to provide more oxygen, and then you can hit it properly with good treatment. Now the question is, what can we learn about blood flow and networks of blood vessels and so on, and what can we do in terms of modeling and simulations? And the idea is really to understand the laws of blood flow from a small scale, and small scale means a single bifurcation, capillary diameter, which is typically the diameter of a single red blood cell, up to the organ level. And as you can imagine, because blood is a suspension of particles, you have strong confinement effects in small blood vessels in particular. You cannot treat blood as an effective liquid that is homogeneous, which means the flow behavior of blood in these networks is highly non-trivial. And that is a figure here that I like to show in particular to an audience when I talk about modeling, because the art of modeling is really about recognizing what kind of physics and maybe chemistry, biology you have to include, and what ingredients can you neglect. So in this particular case, we typically start on the lower left, where we have the smaller blood vessels, smaller flow velocities, everything is in the creeping flow limit, low Reynolds number. Confinement is very large, which means all the blood vessels have a diameter comparable to the diameter of a blood cell. And therefore, you cannot get away without having some kind of model of red blood cells flowing in the geometry. And there are all kinds of models available for that, and I don't want to favor one particular one here. Of course, I will talk about those models we are using in my group here, and also in my colleagues group who is a collaborator on these projects. But you will find other models about blood flow in the literature, and I would say they are relatively equally good if you want to understand what the impact is of blood cells on blood flow. Now, if you move up to the top right, then you can see that the scales increase. You have larger and larger blood vessel diameters, you have higher flow velocities, and blood can be considered more and more as a homogeneous liquid, and maybe even at the larger scale as a single-phase non-Newtonian or even a Newtonian liquid. The question is, how do you know whether you have to apply red blood cell modeling in a given geometry, and when can you get away without? And in particular, if you have an entire organ, you cannot really afford running a simulation on an organ level with all these tiny red blood cells inside. So what we want to have really in the end, and that is the holy grail that I mentioned at the bottom of the slide, we want to have well-informed and reliable macroscopic laws that have in them an understanding of the microscopic behavior of blood cells, in particular in networks. Just a quick comment about the numerical model. I told you it's only a single slide because I wanted to keep it brief in this meeting. We are using a computational method that is lattice-based for the fluid solver. In this case, it's lattice-Boltzmann, but in principle, you can take any other Navier-Stokes solver. You can use finite volume. You can maybe use even multi-particle collisional dynamics, anything that solves your Navier-Stokes equations to a specific level of accuracy. In our case, it's Eulerian. So we have a lattice-based approach for the red blood cells, and we don't consider other cells at this time, only the red blood cells, which are most abundant in blood flow. We have a Lagrangian model, which is based on a simplified finite element model, which means we have single red blood cells, not single red blood cells. We have an ensemble of individual red blood cells moving in complex geometries, and all these particles can deform, and they can go into different child branches at given bifurcations. And it is all about where the red blood cells actually go in a network, because you can create regions of depletion where you have fewer red blood cells than you would naively expect. And in order to get these effects properly resolved, you need deformable red blood cell models. Okay, and for the coupling, you of course need a coupling because the red blood cells affect the fluid flow, but the fluid flow also affects the red blood cells. They get deformed by the fluid stresses, but the viscosity of the fluid of the suspension is changing because of the presence of the particles. We need a two-way coupling, and this two-way coupling is realized by the inverse boundary method in this particular case. But as I said, you can use other methods for that. I don't want to be too specific here. Any method that resolves red blood cells sufficiently well, any kind of fluid structure interaction will do the job as long as the method is properly tested. Okay, so let me talk about the first problem we looked at, and let me be a bit more scientific here. That is actually a work that I've been working on with Miguel Bernabeu, who is on the picture. And I actually forgot to update this slide because this paper has in the meantime been accepted. It's now published in PNAS, where we looked at the effect of red blood cell distribution that is abnormal in an abnormal morphology, which can have an effect on the oxygenation of tissue in an unexpected way. So essentially what is happening, I mentioned that earlier, that the nicely structured capillary bed that you see on the left-hand side that you would expect in healthy tissue, it looks completely differently in a tumor, or it can look very differently. You can have shunts where you have direct connections between arteries and veins. You can have loops. You can have areas that are not well perfused at all. And as a consequence, you can have this hypoxic region where certain tissue is not well supplied by oxygen. Now, the idea is we know from, or the research community knows from observations that tumor tissue is very often hypoxic. That is a well-established fact. And we also know that the tumor vasculature is abnormal. That is also a very well-established fact. Now, the question is, how are the two related to each other? Is it a simple effect that you have regions without red blood cells? And without blood vessels, where you just don't get any oxygen in the first place? Or is maybe something else happening? Is it, for example, possible that you have a network that is so poorly designed that you, although you have blood vessels, you just don't have many red blood cells inside? So the question we wanted to ask is, are there any mechanisms that could lead, other than the absence of blood vessels in certain areas, that could lead to poor oxygenation in tumor vasculature? And here it is important to understand, to some extent at least, how red blood cells distribute in bifurcations and networks. So for example, here you see a double bifurcation. The flow is coming from the left. You have the first bifurcation, and you can see that there's a cell-free layer forming at the top that is relatively large. You have, due to the suspension and confinement effect, you always have a layer close to the surface of the blood vessel where you don't find many red blood cells, and that is called a cell-free layer. And the thickness of that layer can determine, or it determines actually, how many red blood cells go into a given child branch. So for example, if you have a flow coming from the left and you have a child branch going to the bottom, since the entire suspension is affected by that, you may get an increased cell-free layer thickness here, and this will bias the distribution of the red blood cells at the next bifurcation. So the question is, what happens if you have a build-up of this kind of bias in your network? Is it possible that you get blood vessels that are less perfused than you would have it in a normal network? And the most important factor here is the distance between two consecutive bifurcations. So if you imagine you have one bifurcation, it acts as a perturbation to your suspension. But if you have a blood vessel that is sufficiently long, any kind of distortion in the distribution of red blood cells in that blood vessel will recover and will decay after some time. And this CFL, or cell-free layer recovery length, has been established to about 25 channel diameters, right? So if you have some kind of distortion in your flow, then you will see that after roughly 25 diameters downstream, you cannot really see this kind of distortion anymore in the distribution of the red blood cells. In the meantime, we found that this is actually only true for denser blood suspensions at least 20–25% hematocrit, which is the volume fraction of red blood cells. But if you are at lower concentrations, this length can actually be much longer. Anyway, so what we know is that in healthy vasculature, the typical distance between two consecutive bifurcations is about 70 diameters of a blood vessel. And based on everything we know, that is usually long enough for the blood suspension to recover and to forget about any distortion that happened upstream, which means each bifurcation can be considered an independent bifurcation. However, in mouse tumors and other tumors that can be examined by microscopic imaging, we found that the average distance between consecutive bifurcations can be as low as 3 or 4 channel diameters. And after this distance, you can still see the distortion caused by the upstream bifurcation. And the question is, well, what is actually the effect on the suspension flow, in particular on the branching behavior at the next bifurcation when we have this kind of memory? And this is what we looked at in a very simple model. So what you see here is a double bifurcation. Again, flow is coming from the left via branch 0. And we have the first bifurcation with child branches 1 and 2, and the second bifurcation with child branches 3 and 4. And as you can see, we have varied in this simple model the distance between the bifurcations and also the relative orientation of the bifurcations. We used typical parameters, 20% hematocrit, which is quite normal for the microcirculation, a diameter of about 33 micrometers, and a typical flow velocity. And now the question is, what happens to the flow split? Now, we set up the simulations in a way that the flow rates in branches 1 and 2 are the same. And by flow rates, I mean the suspension flow rates, the total flow rate of the entire blood suspension. And also in 3 and 4, they are the same. So we have 50-50 split of the suspension flow rate at the first and at the second bifurcation. Now, if you look at the hematocrit, the volume fraction of red blood cells in the branches in the first setup where we have a sufficiently large distance between bifurcations, it's about 25 diameters, you can see that statistically speaking, the first two branches, 1 and 2, they have a basically equal suspension concentration. the red blood cell concentration, hematocrit is roughly the same. And if you go to the second bifurcation, you see that again, the split of red blood cells equals the split of the suspension rate. Now, the question to you, think about it for a second. What do you think will happen when the bifurcations are closer to each other? Ask yourself, will you get the same behavior, or will you have different concentrations in branches 3 and 4? The answer is that you have a significant difference in the concentration in branches 3 and 4. And depending on in which direction the branches go, you get an increase or decrease of hematocrit in branch 3 or branch 4. The essence here is we have an asymmetric red blood cell split. And that is really important because we know that this kind of effect can be magnified after consecutive bifurcations if the bifurcations are very close. And in the human vasculature, you have multiple generations of bifurcations. If you want to look a bit closer to see what is happening here, then you can see that flow again is coming from the left. The red blood cell distribution is relatively equal when it comes to the bottom and the top wall. Then you have the first bifurcation where you have an equal or an even split. And if you look very carefully at the segment between the two bifurcations, you can see that the red blood cell suspension has been shifted upwards a bit. So the cell-free layer at the bottom is larger than at the top. At the top, it's basically non-existent. And this cell-free layer asymmetry does not have enough time here or enough distance to recover, and that causes an uneven split. And in fact, the branch going up in the second bifurcation has a higher concentration, and the branch going straight in the second bifurcation has a lower concentration. Also, what we did, we proposed an extended model based on the pre-second model that is already 30 years old. The original PRES model is able to predict that red blood cells can actually split in a way that doesn't equal the split of the suspension at a bifurcation, but that would only happen if the flow rates are not equal at both branches, child branches. We found that we can have an unequal split of red blood cells even if the suspension flow rates are equal in a bifurcation. And I don't want to talk about the mathematical details here. We suggested an extended model that can capture that kind of memory effect. And based on that, we actually investigated also in a simple model the oxygenation in a network, and we found that this effect can lead to heterogeneous oxygen distribution in the network. And that is essentially what happened in the PNAS paper. I do not want to talk about all the details here. If you are interested, you can take a look at the paper. The key takeaway message here is that due to suspension effects and memory effects, you can have an unexpected and asymmetric distribution of red blood cells, and this effect can accumulate because you have a hierarchy of bifurcations in short distance, one after the other. Now, the second application I want to talk about, that is you can see here Charles, who is, he was a PhD student, is now a postdoc in my group. And he looked at the role of red blood cell split in the developing retina. So the question is, if you have a network of blood vessels, these networks, they are not fully developed when you are born, for example. In the retina, this is particularly obvious, where if you look at the mouse retina, for example, you can see that over time it still develops. It takes about 3 weeks after birth until the network of blood vessels in the retina has completely developed. And the question is, how does it actually develop? What are the cues? What are the triggers that are informing the biology to form a proper network? And we believe that feedback due to the flow of the red blood cells is crucial. So it's not only, it's not that the design of the network is inherently known by the genes or something, but there must be some kind of flow regulation and suspension regulation that leads to the formation of a proper network after some time, after about 3 weeks. So what we did, we took the image of a retina. You can see in figure A, a mask we got from experimentalists who gave us this data. Then what we did, we created out of this two-dimensional network, we created a three-dimensional network. The retinal blood vessels are essentially in a plane. It's in a curved plane on a hemisphere, essentially. But locally, it is roughly flat. So you have a two-dimensional network, and we assume that the diameter of the blood vessels is circular. The cross-sectional shape is circular at any point. So based on this two-dimensional network, you can therefore create a three-dimensional geometry. Then we ran a simulation without red blood cells because the geometry is actually a bit too large in order to run red blood cells through the entire geometry, just to get a feeling for where the inlets and outlets are in specific regions of interest. This is what you see in segments D, E, and F, where we picked a smaller subvolume. And for this subvolume, we know now, based on the bigger simulation, what the inlets and outlets are. And now we run simulations with red blood cells. And let me show you a video, which you should be able to see now. So we have three regions of interest I'm showing you here. And you can see red blood cells moving through this network. And you can see that there are some branches that are not well perfused or not even perfused at all. And if you look carefully on the left, you have a branch now that is blocked because a red blood cell is stuck inside. The idea is now, is it possible to somehow link the perfusion, which means the amount of red blood cells going through, with the development of the retinal network? So what I want to, this is now, okay, this is difficult to explain. How can we actually look at the development over time in a retina? What you need to know is, in order to image the retina, our experimental colleagues actually have to extract the retina, which means you have only one shot. You can only see the retina at one instant in time, and you cannot track the same retina over an extended period of time. So how do you get information about the time development of the geometry anyway? The idea is that you look at different biological markers in the same geometry. You can see at the top left, there are two different markers. One is in green and one is in brown or in red. One is for collagen and one is for the endothelium, which is lining the inside of the blood vessels. The collagen is basically showing you the structure around the blood vessels, and the endothelium is showing you the inside. Now, what is happening? It is known that some of the blood vessels are pruned over time and they disappear. And it is not really that clear why that is happening. So we want to know, why do these blood vessels disappear over time? And the idea is here that blood vessels that have the collagen marker, but not the endothelium marker, these were blood vessels in the past that are disappearing, and the collagen matrix is still in place. So essentially what is happening, the endothelial cells, they usually make the first move. If you have a new blood vessel forming, the endothelial cells start forming a blood vessel, and after that the infrastructure gets in place. And if a blood vessel disappears, the endothelial cells decide to migrate elsewhere. They go away, and you leave with the structure of the blood vessel, but it's not there anymore. So you have the collagen matrix around it still, but the endothelial cells have moved on. They are elsewhere in the body. They are recycled, basically. And now the idea is that blood vessels here that are both green and brown are fully perfused blood vessels that have also been perfused in the past. Blood vessels that are only brown have been blood vessels in the past, but they disappeared in the meantime for some reason, right? And we want to know why. So what we did, we ran simulations in the old geometry. So we used the collagen marker for our simulations, and we correlated the perfusion of the red blood cells with the fate of each blood vessel. We looked at, is there anything specific about the blood vessels that later disappeared when we look at the red blood cell perfusion? And indeed, we saw that those blood vessels that are well perfused are those blood vessels that exist later on, while those blood vessels that have hardly any red blood cells inside, they disappear. You might ask now, how do you know, if you just look at the network, can you tell which branches have more and which branches have fewer red blood cells inside? And actually, you would say, well, it's the diameter, of course. So the diameter tells you how many red blood cells can go through, but unfortunately, that is not correct. This is, of course, what we thought initially, but this is not really what we see. In the left diagram, you can see that the horizontal line for Q RBC minus Q blood equals 0, this is where you get the same fraction of red blood cells compared to the feeding bifurcation when you look at the red blood cell split and the plasma split. So essentially, if you are on the horizontal line, then you get exactly as many red blood cells as you would naively expect based on the flow split of blood as a whole suspension. Whenever the black dot, each black dot is one blood vessel, whenever the black dot is in the negative, it means you have a depletion of red blood cells with respect to the plasma. And whenever the black dot is positive, it means you have an enrichment of red blood cells in a given branch. And this is shown as function of vessel diameter. And you can see, except for the very, very small blood vessels, which are 4 micrometers, which is the radius of a red blood cell, they are tiny. If you forget about these very small blood vessels, there's no clear correlation between enrichment or depletion of red blood cells and the actual diameter of the blood vessel. However, what we see is that it is actually the flow rate itself that determines how many red blood cells go into a vessel. So essentially, if you have a branch that doesn't get a lot of blood flow in the first instance, then it will get even fewer red blood cells than you would naively expect. So essentially, maybe I can show that before I conclude. At the top left, if you have a suspension, it's a two-phase system, right? And if you have now a splitter, a bifurcation, I mean, the typical chemical engineering example, a splitter doesn't change the composition of a mixture. This is what you teach your students. But this is unfortunately not true for blood flow, because at least on the microscale, because you have confinement effects and the red blood cells behave differently than the liquid. So you can have situations where the branch that receives a bit less suspension will have even less red blood cells, or vice versa. If you had a perfectly naive split of red blood cells at a bifurcation, you would follow the diagonal dotted line, which means if you get 50% of the blood as a suspension, you would expect 50% of the red blood cells. If you get 25% of the blood, you would expect 25% of the red blood cells, and so on. What we see is, this is actually a known effect, but it hasn't been described on this network level, and in particular in connection with the blood vessel development. What we see is that blood vessel branches that receive a very small amount of blood as a suspension, they hardly get any red blood cells, even less than we would naively expect. And those blood vessels that are well perfused, they get a very, very large amount of red blood cells. And that is actually the predicting factor. And based on that, we can then say which blood vessel will later on disappear. Unfortunately, if you look at the network as a whole, you cannot simply tell which branch will disappear, because you need, first of all, the flow simulation of the suspension in the network in order to get that information. So we are currently not able to look at the network as a whole, the topology, the geometry, and predict based on that how the blood vessel network will develop. So that is work in progress. Now, there's ongoing work. We just submitted a paper where we look at the effect of a compression on the split of red blood cells in a bifurcation. This is what you see at the top. I mentioned at the beginning that tumors can compress blood vessels, and we have seen that this kind of compression, where the diameter of a blood vessel changes just before bifurcation, also leads to a bias in the distribution of blood cells, and this could contribute further to the oxygen depletion in tumors. And also we have a recently funded EPSRC project where we look at the distribution of red blood cells and blood flow in the human placenta to understand better how the geometry of the placenta is linked with diseases of the placenta. In summary, the ultimate aim, this is really what we want to get out of this entire discipline in the future, is to have well-defined effective blood flow models for the macroscale that are applicable to entire networks where you just cannot afford running detailed simulations. It's basically like coming up with a Navier-Stokes equation from MD, where you get an expression for the viscosity, right? The viscosity is a macroscopic concept. It doesn't exist on the microscale. But for Navier-Stokes, viscosity works beautifully well. We would like to have something similar for blood flow, blood as a suspension of red blood cells on the network level, where we have a model for the uneven and biased split of red blood cells in bifurcations. But this, of course, requires that we understand the mechanisms. And this also means we need to run more of these simulations and develop better models that capture that kind of behavior. So the cell level modeling is still crucial. I'm not advocating the removal of cell level modeling. It is definitely crucial in order to develop the macroscopic models that we are so much interested in. Thank you very much for your attention. I think it was about 30, 35 minutes.